Hello, we are continuing on with passive transport right now. The first thing, kind of passive transport that we did was diffusion, and number two now is osmosis. Okay, so here is a definition for osmosis: diffusion of water across a membrane from high to low concentration. Okay, again, high to low concentration. This does not require energy because it's passive transport. Water will naturally go from where there's more concentration of water to a less concentration of water. And the reason our cells need to do this is to keep a water balance inside and outside of the cell, the proper water balance. Remember, this keeping a balance of different things is called homeostasis. So again, homeostasis is what we're trying to do here. And this cell right here perhaps was in an environment where water would go into the cell, and then it ended up getting too full and then kind of exploding out here. Okay, so we'll kind of talk about some situations where if cells are in different environments, that could be detrimental to them if they can't maintain a proper water balance. Okay, so let's continue. All right, so here we have let's say this is a beaker, and down the middle is this semi-permeable membrane. Okay, so only water. So these little guys, the blue dots, obviously, are the water molecules. The red dots, let's say, are sugar, and they cannot go through the membrane. Okay, so we're going to compare the two different sides, right? So on the left side, we have a high sugar concentration. Okay, and that means, since we're comparing it to the other side, if there's a higher sugar concentration, that means the other side has a lower sugar concentration. And we can see that with the fewer dots here. Okay, so what does that mean about the relative water concentrations? If we have a lower sugar concentration on this side, then that means this side also has a higher water concentration than we have on the other side, right? Higher and lower on this side. Okay, and that should make sense. Everything here, if we have a higher sugar concentration on one side, it has to be lower on the other side, which means that we have a higher water concentration on this side, which means there must be a lower water concentration on the other side. And all of those things that I'm saying is backed up just by the picture here, right? So now we're talking about osmosis, which is the movement of water from high to low concentration. So we know that it's higher on this side and lower on this side. So these water molecules that can go through the membrane are going to go over this way in order to even out as much as it can the concentration of the sugar molecules on both sides. Okay, so it's going to try to even it out. It's kind of like on this side, you have very concentrated Kool-Aid, and on this side, so it's very sweet, and on this side, you have less concentrated Kool-Aid. And so what's going to happen is the water here is going to go to this side to kind of dilute the delicious sweet Kool-Aid and make it more like itself. Don't know if that was great. I think it makes sense, but don't know why anyone would want to do that. All right, so what would happen in this situation here, if we have water goes from a high to low concentration, is that all of these water molecules, when they move across, they would move over here, and then this water level might raise if we have this kind of setup in particular where like the water can go up on one side. Okay, so the second picture here is just showing the effect of water moving from the right side where it was high water concentration to the left side, which is low water concentration. Okay, that is osmosis. Let's continue. Now, we have to learn some important terms for osmosis, okay? And so I'll just remind you here, solute is the stuff that we dissolve in a solvent and we make a solution with, okay? So the terms that I'm going to go over now have to do with how much solute you have in your solution. So we got to write these down. These are very important. I'm going to use these when we're doing our egg lab. So hypertonic, a solution with a relatively high amount of solute. Hypotonic, a solution with a relatively low amount of solute. Okay, so we can kind of remember that with the words hyper and hypo. Everyone probably is familiar with the term hypothermia. which is when you are very cold, right? When your body temperature drops below what it should be. So hypo means kind of less. So here with hypotonic, we have a relatively low amount of solute. So the amount of stuff that's dissolved in our solution is lower than something else. Hyper means more, okay? Hypo means less, hyper means more. Some of my students like to say, if you have a lot of sugar, you are hyper. You need a lot of sugar, a high amount of sugar. Okay. The last word that you need to write down here is isotonic. Okay. A solution with equal concentration of solute as another. Okay. So it is the same as something else. So this is more than something else. This is less than something else. And this is the same as something else. The same amount of solutes. Okay. If you were to guess, about like what one of these words should describe your blood concentration compared to your cell's concentration, hopefully you would guess isotonic, right? Most of the time we want an equal concentration of things on either side. Not always, but depends on this specific case. Okay, so let's continue. And I want to just kind of really emphasize this term. So it says, what is wrong with these comparisons? This car is faster. The man is hungrier. The cell is bigger. Okay, the cell is hypertonic. So these don't make sense. Hopefully you get this, because compared to what, right? You have to say the car is faster than this other car. This man is hungrier than this other man. Okay, just like we can't say the cell is hypertonic, we would have to say the cell is hypertonic compared to something else, right? So when we look at these pictures down here, we have the cell, and then outside of the cell, okay? And so we are always going to compare this outside of the cell to what's going on inside of the cell, okay? So these are the the two solutions we are comparing, the solution from inside the cell and the solution outside the cell. So in this case, we have hypotonic, which means there are fewer solutes outside than there are inside, okay? There are fewer or less solutes out here, okay? On this side, we have hypertonic. So you can see out here, they have a lot more solutes, or remember, things dissolved, than we have on the inside, okay? So that is hypertonic. And then the solution, not the cell, the solution. Oh no, go back. The solution is, okay, well, this is struggling. The solution is hypertonic, okay? Last one is isotonic. So we have roughly equivalent solutes inside and outside of the cell, okay? So same, fewer, more. But when we use these terms, we have to be comparing two things. Otherwise, the words don't work. All right. So let's go through some examples. So I want you to look at this, and let's say these orange ones are sugar molecules and these ones are water molecules. It says, which of the following best describes the solution surrounding the cell? So we're talking about out here, not the cell. Okay, so the solution has, I would say, roughly the same number of solutes inside and outside. So we'll say that it is isotonic. Okay. So which direction will water flow? Well, let's say water goes in. Well, that's going to make it a little bit crowded. So maybe some water then goes out to even it out again. Okay. So in an isotonic solution, water is going to flow both in and out. Okay. Kind of at an even rate. Next. Okay. So which of the following best describes the solution surrounding the cell? Well, we have kind of a lot fewer of these solutes on the outside than the inside. The inside is a lot more concentrated with these orange dots. So that is going to make the solution out here hypotonic because there is less, okay? And that means we have to look for which way the water is going to flow, and there's a lot higher, if there's a low solute concentration on the outside, that means there's a high water concentration. So that means the water is going to go from outside where there's more to inside where there's less, okay? So into the cell, because remember, water goes from high to low concentration. So good. Next one. All right, which part of the which of the words describes the solution surrounding a cell? Here we have the opposite situation. We have a lot of orange dots on the outside, which may be a sugar or salt or something. So it's high concentration of solute on the outside, and that means that on the outside in that solution we have a low concentration of water. And the opposite is going to be true for in here. We're going to have, so you can see there's fewer solutes. So I'm going to say low concentration of solute, and that means there's a lot of more water, high concentration of water. So water goes from a high concentration to a low concentration. So the water is going to leave the cell, out of the cell. What we said, the outside is hypertonic. Okay. So I want you to understand these situations. Probably draw a couple of these situations where you can draw dots and show which way the water is going to move, into or out of the cell. Okay. I want to leave you with this final thing here. We just went all over all of this. I'm not really going to talk about it too much. This is just a nice organizational tool here. So if you want to take some notes from this or draw this or take a picture of this, that would be great for you to have. Okay. So let me know if you have any questions about osmosis, and see you next time.